Okay. Maybe. Okay. For insulin to have any effect, it has to find its receptor. And just to remind you, this is the cell membrane. And here's the insulin receptor, made up of 4 subunits: extracellular, alpha, transmembrane, intracellular, beta subunits. All 4 are part of the insulin receptor. When insulin comes from the bloodstream, it will bind to the outside on the extracellular surface, to the alpha subunits, and activate the intrinsic tyrosine kinase activity of the receptor. This is a bit of a review for you guys if you've forgotten. Here's the insulin hormone, this heart-shaped structure, binding to the extracellular surface of the insulin receptor. In doing so, it is going to activate the enzyme activity of the beta subunit. The enzyme activity of the beta subunit is a tyrosine kinase. What does that mean? That means that the beta subunit can phosphorylate tyrosine residues first on itself. So I want you to kind of think about this and understand it. This is a polypeptide, the beta subunit, with many tyrosine residues as part of that polypeptide. So the first thing that happens to the receptor is to autophosphorylate, converting specific tyrosine residues in this beta subunit to phosphotyrosine. That's what the P. So that's a sign that the receptor is activated. The enzymatic activity of that activated receptor can also phosphorylate tyrosine on other cellular proteins. And these could be target enzymes, these could be adapter proteins. There are multiple substrate proteins that can be phosphorylated by the insulin receptor tyrosine kinase activity. Okay? And that is essentially the activation of the tyrosine kinase activity of the receptor initiates a cascade of events that results in the cellular response. Say insulin is going to activate glycolysis or glycogenesis. That's what it starts. That's how these cellular responses are initiated, by first activating the tyrosine kinase activity of the receptor. Okay, so here's a little summary, if you want, of some of the important substrates for the tyrosine kinase activity of the receptor, the insulin receptor. Obviously, first it has to autophosphorylate, meaning phosphorylate specific tyrosine residues that are part of the beta receptor. These aren't free tyrosines. These are part of the receptor or part of specific proteins in that primary sequence. So there are many known substrates. They're called insulin receptor substrates, protein substrates. There are some adapters, proteins, and some enzyme proteins that get phosphorylated. So we're not going to get into the detail, but all of them get phosphorylated at tyrosine residues, and that propagates the cascade that results in a physiological response. So here are some of the important responses that insulin will elicit. insulin will increase glucose uptake in specific tissues. Okay, we'll talk about that in a little bit, some more, in some detail. Insulin can regulate the transcription of specific enzymes. Insulin can upregulate, for example, glucokinase, PFK1, and pyruvate kinase. It can result in the covalent modification by dephosphorylating certain enzymes. For example, in glycogen synthesis, because this is recent, insulin promotes the dephosphorylation of glycogen synthase, activating it. So many effects of insulin, including regulation of transcription, glucose uptake, and covalent modification of important enzymes. And that is the underlying cause or a reason for some of the insulin effects. So I'm going to talk a little bit about how insulin increases glucose uptake, just as an example. And it's really important that you understand that. So here's the receptor here in blue on the cell membrane. Let's say this cell is a muscle cell. Okay? And here's insulin binding to its receptor on the extracellular surface. The first thing that's going to happen, as we said, is to activate the tyrosine kinase activity of the receptor, which will then phosphorylate other protein substrates in the cell, generating this cascade of events. Okay? One of the important results of that cascade is that you see here the Golgi apparatus. These are Golgi vesicles. They contain this sort of donut-shaped structure. White donut-shaped structure is the GLUT4 transporter. So insulin, through activation of the receptor tyrosine kinase, generates this cascade of events. We won't get into the details, but promotes the translocation of these vesicles to the cell membrane, fusing with the cell membrane, thus increasing the number of GLUT4 transporters in the cell membrane. If you increase the number of transporters on the cell surface, you're going to increase the uptake of glucose from the bloodstream. And that's exactly what happens. And this is really very important. Insulin has this effect on only two tissues: muscle, including skeletal and cardiac, and adipose tissue, because those are the only two tissues that have the GLUT4 transporter. Insulin does not have these effects on the remaining tissues in the body, only on these two tissues. So uptake of glucose is increased in only skeletal muscle and only in muscle and adipose tissue. Okay? Because they have the GLUT4. So this is a unique effect on the GLUT4, an increase in GLUT4 on the cell surface of these tissues. And that's really the same thing, but just to remind you that everything starts by activating the tyrosine kinase activity of the receptor here, where it autophosphorylates, and then eventually it will phosphorylate other cellular proteins and propagate the cascade, that one of the cascades that we just talked about, like the increased glucose uptake. And that's the same thing as well, simpler. This is the vesicle here, activation there, activation here, sorry. You have a cascade of events here that eventually, through tyrosine phosphorylation of unique proteins, that initiates the translocation of Golgi vesicles, increasing the number of transporters in these cells, muscle or adipose tissue. When insulin dissociates, that is terminated. Its effects are terminated. Okay? And that's really done by dephosphorylation of the receptor eventually, and that terminates its effects. Dissociation of insulin and dephosphorylation of the receptor. So this is just a reminder to tell you that really most tissues in the body are, or transport of glucose in most tissues in the body is not insulin dependent. Only skeletal muscle and adipose tissue glucose transport can be increased by insulin. And that's actually quite important to appreciate. Muscle, because of its mass, can pick up a lot of glucose and use it for its energy source, for its energy needs. So can adipose tissue. I'll give you sort of a real life importance of insulin. In diabetics, let's say diabetics type 1, whose pancreas cannot produce enough insulin. Because they cannot produce enough insulin, the uptake of glucose in adipose tissue and muscle, which constitute quite a bit of mass, body mass, is actually decreased or not increased from the bloodstream. What does that mean? That means glucose accumulates in the blood, because these tissues aren't picking up glucose because of the absence of insulin. And that's what results in hyperglycemia and eventually diabetes. Okay? So that's the importance of this, that insulin promotes the uptake of glucose from the bloodstream, thus maintaining blood glucose in a certain range in the blood. If it's not released, because say in diabetes type 1, then that glucose is not taken up by these tissues, and it will accumulate and really result in hyperglycemia and eventually diabetes. Okay. If you like this channel, please subscribe. Thank you.